Right. Let's kick off. Hi, everyone. Welcome back. I realized at the start of the day I didn't introduce myself. Oops. So, hi, I'm Katie O'Connor. I'm a senior strawberry breeder in the Department of Primary Industries in Queensland. I'm also an adjunct senior research fellow at UQ. Okay, so for the next little block, I'm so excited. We've got Dr. Luke Yates from University of Tasmania, Nipaluna, talking about modelling development, architecture and yield in garden pea with AI-assisted Bayesian inference.
Is this microphone working? Can you hear me through the microphone? Yeah. Okay, good. Okay. So I guess a little bit in terms of AI, we've been talking a lot about different ways it can be used, and I guess what I want to talk about today is using AI to help you do classical things. So I'm interested in Bayesian inference, and I'm interested in constructing models where I'm not just wanting prediction. Prediction's great, but I also want to know what drives that prediction. I'm interested in what are the causal factors? Can I construct networks and things that help me unpack really what's driving something? And I think these are at the heart of scientific questions, from the beginning of time. How do we separate causation from correlation? Is our goal to explain or to predict? And I think we're often caught between these worlds in a lot of what we do. And classically, we often are interested in things like confidence or credible intervals, not just what is the estimate, but how certain are we in it? Can I use that to test the hypothesis that I have here? And data are always annoying, right? And so that's because they're really independent. They're not really what you want. They're structured because of the way you sample them. They're structured because of pedigrees or phylogenies or space or time. And so these are still frontier issues in machine learning, is how to correctly account for these dependencies so that you can make inferences or at least have well-calibrated predictions. And by calibrated, I mean that your 95% credible interval of your prediction would cover 95% of the held out data or something like that. So these are ongoing challenges. The simplest thing you can do in classical modeling is a linear model where you've got some predictors, X, and you've got some coefficients. People call them slopes or effect sizes, beta. And you just have some simple model for the residuals. Anything left over is assumed independently, normally distributed. That's really good enough. You usually have to add some sort of random effects. All that usually means is that you acknowledge that your data have structure, that structure makes some observations more similar than others for reasons that aren't in the predictors you already have. And so there's many tricks and techniques for using random effects to deal with the sorts of dependencies that I've talked about. The other thing you probably aren't happy with a model like this is things are really linear. So you might think, all right, I'll try and make my X a bit more complicated. So now I allow myself some more interesting function, maybe just a known mathematical function or maybe a spline or something like that. And there'll still be parameters associated with that beta, but they might become harder to interpret. And then we come on to machine learning, where a lot of effort is put into F. It's sort of known that neural networks are basically universal approximators. So it's very good at finding F. If you want to find a very complicated function that relates all of the input variables that you have, it's very good at that. And the weights are now the parameters being estimated. But usually what happens, you see from the previous equation to this one, we've lost our random effect structure. We've lost any knowledge or about the dependencies that we care about. And the reason that's important is because if I go back to my first model here, I want to make inferences about beta, the effect sizes of say, traits that drive yield, or effect sizes of how treatments affected outcomes. And getting the correct inference that is, if you're calculating a p-value or a posterior, getting the correct inference depends on having the random structure about right. So we know that things go wrong if you don't do that. So that's my kind of, a prologue or something before I begin to sort of say this is sort of the things I'm thinking about as I work through this. So I'm going to talk about a model for yield in P. And yield, as we all know, and my background is more in modeling and mathematics across biology and ecology than in crop modeling and things. So that's my caveat. Obviously, yield is not an isolated trait, and traits exist in coordination, sometimes called syndromes. And that's because, of course, of their evolutionary history, the types of environments the types of trade-offs that were required. And some of those relationships between traits, you could say, are kind of strongly coupled. Maybe because for biophysical reasons, you just can't allocate energy infinitely. You must trade off in decisions. Other times they may be constrained, let's just say, because of chance of the evolutionary path, that things have become coupled, but they could be decoupled in principle. So for our model for yield, I'm going to be thinking about a whole suite of traits, not just yield, as I try and make sense of some data that I'll tell you about. So for this case study, we worked with a pretty extensive experiment in pea, where peas were grown from seed to senescence. And first of all, there was experimental treatment. So there were 2x2 light conditions. So the far red was about the spectrum of light, and the PAR was about how much light they were receiving. And there were 15 different genotypes with about 8 replicates for each in each condition. And they were measured in all kinds of ways across the time series of their development, particularly at these 4 sort of developmental stages, and a whole suite of measurements, some of which I've sort of written at the bottom there. And so I've sort of talked about it before, but the merits of a trait-based approach are that you can start to unpack, hopefully, some of the causal chain. Things that happen earlier in the development around the timing or around architectural decisions may impact on the yield that we're sort of primarily interested in. That could be helpful because you might want to make predictions from early signals if you're looking at a young plant. Of course, it's also of interest because we're looking at what traits have genetic variation across our genotypes and how they relate to the yield at the end. So it's not so much a science talk in the sense that I do have some results to show you, but I kind of want to talk about how we've used AI in this process, since that's what we're here to talk about. So a word we heard a lot on the first day is context. And for AI, context has a different meaning a little bit to what we mean, but really it's sort of the same. Like I work a lot with biologists, people who are really specialists in their domains, and I come in with my specialities, and the hardest thing is to find that context. So, how do we speak the same language? When I say this, what do you hear? When you say that, what do I hear? Because it takes a lot of time to unpack those things. And once you get that working well, then you can really work together. And it's not a lot different with AI. If you just give it a quick prompt and hope it's going to be great for you, you haven't given it the context. You have to work just as hard with AI to create context. And when you do, I find it very surprising what it can do for you. So that's the sort of thesis here. So I'll talk about the first meetings we had. So we are 2 biologists and 2 mathematical types. And we met a few times and we just had long conversations. They told us a bit about what they'd done, what they're interested in. We grilled them. What do you mean? What does that word do? Why this? Why that? And then the only thing, we weren't using any AI at this point. Those guys had some slides and pictures they'd drawn and things like that. We started drawing DAGs and other diagrams that we thought are going to help us do modeling. We didn't record it with AI, but I wish we'd had, because it would have been very helpful context for AI itself in the subsequent steps. But that's how this one rolled. So just a little bit of kind of fundamentals, perhaps, in working with AI as a sort of assistant on your computer. I've been working with Claude Code, so I'll just tell you about that. But I'm going to help with the workshop later this afternoon if you're attending, and we'll talk about some other options at that point. So I'm just going to show you now. I really hope this works. Yes, good. Just going to show you this is my directory for the P project. It looks a bit messy. You can see there's all kinds of folders and things in there. And this is a fairly mature project, so I've had to create a lot of folders to organize my things. But if I look in the docs folder, I've got illustrations that were drawn by one of my collaborators. I've got all kinds of things that I thought might be useful for the AI. It could look at these images, it could help unpack things, it would help me convey what I really wanted to do. And so I'll just show you here too, just so we're sort of in demo mode. Claude Code runs in the terminal, so I'll open a terminal here. Now I can make it a little larger, I think. So I've launched Claude from in this folder. So it sees everything in that folder. It sees all of my directories. It sees all of the work I've done. It can read anything. And it keeps its own little memory file about what's going on, what it's been asked to do already, what are the important lessons that have been learned, so it doesn't have to keep reinventing the wheel. So you can see straight away, just in a few seconds, it's come through and it's told me a bit about what I'm up to. some things you can do here in Claude. This is picking up on some stuff from earlier. I can choose which model I use. So here I've got it set on Opus, which is the most expensive token using one. Well, nearly. I guess Fable uses more. But you can select which model quite easily. So Haiku is nice and quick. It'd be great for a demonstration in front of a big group when you don't want to wait too long. You can also change the effort. So here between different amounts of effort, which is sort of how long it will think to do things. Now I find in the sort of stuff I'm going to show you, writing complicated models, I like a lot of effort and I like to use a harder model. I'm quite happy to wait a while for things. It's definitely worth the wait. So be slow building context, being slow getting the conversation going, and be slow and waiting for things. That'll end up being quicker in my experience. So I'm just going to come back and forth between this view of what's happening in my folders and my slides as we go along. So, step number one is always, from a modeling point of view, having to deal with the data, how it's been given to you, which is never how you would like it. So if I show you in this data directory, you can see all the iterations of the data because of all the data entry errors we found. And I'll talk about how we use Claude to find those. But if you look in that, you can see there's folders with different time points in there. I've got bundles of Excels. Each of those have different sheets. That's a very natural way to organize it if you've gone and collected that data and you've done all those things. Of course, it's not a natural way to model the data. So it's not a trivial thing to sort of aggregate correctly, 40 odd Excel documents with sheets and not make any transcription errors or mistakes or things like that. It's quite hard to do well. And, we can spend a lot of time doing that. So I want to show you to get Claude to do that. I'd already had the conversations, the sort of get to know yous that we talked about already. And then I asked it to sort of create a script that would condense all of this down to just 3 CSV files, the sort of things I want by node, by plant, things that are independent of time, that sort of thing. And I also asked it to make a nice HTML document about how it did it. So it's gone through and it's written it like a tutorial, which is I asked it to do. So it tells me about the experiment. It says, yes, you do have a lot of files. Here they all are. Look at some of the issues it found, spellings in column names, these sorts of things. It writes them all down for me. And then it talks about what the schema is going to be in the new sheets. So I've got that for my various ones. And then it actually, all below, it shows all the functions and things it wrote to do the wrangling. And I can read through it and it's very impressive. I read through it and think, oh, that's such a nice way to do that. Oh, what a great idea. I can easily be happy that this is at least as good as something I could manage with a couple of weeks of anguish. So and then I keep that. So that's now one of my docs. That's another resource for Claude. It can always remember what the data was and what it's done. And then also, so following that, we made a data exploration. So, okay, I've got my nice CSVs. Let's look at the data. And there's a lot of ways to, you can see all the code has been printed here that Claude's generated, and it's made many, many, many plots about the data. Now, I'm not an expert in the field, so I look at the data and think, oh, looks right, I suppose. There's the odd point that looks a bit wild. That's not for me. This gets straight back to the biologists. They look through this data. They grew those plants. They know their species, and they straight away go, oh, that's not right. Well, what about that? And they go back to their notes. Okay, it was written correctly in pencil. Oh, it's not correct in the Excel sheet. Such a common thing. We actually were able to find a lot of errors through this visualization. And as I'll show you, we found even better ways to find, possible issues with our data entry. Okay. So I'll talk a little bit about the model. I mean, this is sort of my domain more. I've got a lot of traits at hand. Let's say I think we've got about 12 to 15 in this first model. And as we just heard about in the previous talk, a multivariate approach. I'm trying to think about how these traits themselves are modeled independently, but also how they're correlated across the data set. So I just want to take 2 traits to sort of get us thinking along the lines of how you might start to build such a model. And so early developmental traits, so I've got days to flowering and the node of flowering initiation. Now, these are both just positive integers in our data set. So a negative binomial is just a distribution choice, which is pretty good for integers, especially if you're not too sure how variable they'll be. So here I have the basic model. I say, okay, my days to flowering is going to have some sort of mean and some sort of dispersion. It's completely general at this point. But already I have to think for the next line, node of flowering initiation. Well, I can get that node if I know how long it took to get to that time and how many nodes it made per day. So I'm already thinking there's a relationship between the previous model and this one. And so now I can also unpack things. So when it comes to ask, I'll put this next line up. When it comes to if NFI is higher for a given genotype, I can ask, was that because it took more time or because it had a faster node production rate? I can start to unpack this small part of the model here. Now, for every model, every distributional statement like the two I've got there, I'm going to have some mean that I'm estimating. And for that mean, I want to ask, well, did the light conditions make a difference? Did they affect that trait in that part of the chain? And then what was the role of the genotype? Were the genotypic differences, were there high genetic variance in that particular trait? So these are kind of classical things you might do. This is a random effect, and that little A just means I'm going to correlate genetic effects across the different traits that I'm modeling. Now, to work up to more traits, I mean, I could write that model myself by hand pretty quickly, but this one, I got a lot of traits. So I started drawing these diagrams with Shane, my collaborator. We'd sort of think, well, here are some traits. And that little box, we said that box, you can see the notation at the bottom, stands for light conditions plus genetic effect. And Claude when we scanned this in, and he saw the box, he goes, I get it. You want a box on everything. Yep, good. And start using the word box for us, in the conversation. So we know you're carrying around, this unit of measurement. Then we thought a bit more ourselves. I'm not going to run you through this one, but just to show that for each trait, we thought about what's the good distribution, what's the right relationship, and tried to think for ourselves around the biology and make sure things were linked in natural ways. Now we move towards AI a bit more. We say, well, okay, draw us a little diagram to show the chains of connections between these traits. So you can see it's drawn this sort of simple DAG. Here, a directed graph that is. And then we said, okay, that's good. We want to write this up. Now, there's not software that'll just fit a model like this with all these different sorts of traits. So we use Stan, which is a programming language for Bayesian inference. And we said, oh, look, use Stan. We had some example models we'd written in the past. Here's some code that we liked. This is our kind of conventions. And it was very, very quick. After grilling us a lot about some of the errors in our previous spec document, which we resolved, it then wrote this lovely piece of code. Now, most people wouldn't find this very beautiful, but I thought it was very quite nice. The way it was lined up, it's very elegant, and it made it look quite simple. And I thought, oh yeah, that's quite nice. I can read that code, and I'm happy for you to fit that. And so I just want to keep an eye on the time because we did start late. What time am I meant to finish? 5 past. Okay, I'll keep moving. Good. So I'll show you some initial results. Like I said, I'm not here to really tell you about the scientific results, but I will show you some of what we got. So looking at the light conditions, so this was sort of you can see all the traits are on the bottom there. Just to pick one to look at, this one here is the days to flowering. You can see that there was a reduction in days to flowering under the high par scenario, so the high par light condition. And in fact, the par was very, very important across the board. It would make quite a big difference to various of these traits. Good if you're interested in experimental design, good if you care about the light conditions, but we're not really leveraging our data by looking at these plots, or our model that is, because we know a lot more about the connectedness of these traits, and we want to exploit that. So that's great, that's already interesting. So on this next plot, and now this one here, now it takes a moment to unpack this one. So the traits are on the bottom of this x-axis here. Now they're a little bit hard to read, I admit. So first of all, the thing to look at is that there's definitely patterning. There's more or less two classes of genotypes. And if I took this as an example, the second one here, which is the number of days it spends in the reproductive time, so from it starts flowering to it finishes flowering, there's ones that spend a lot longer and there's ones that spend a lot less. So that was interesting observation straight away. And you can actually see, if I start to highlight some of these, ones that are high there tend to be lower down in this next one. So this, if I go right over to this, it's very hard to keep the color right. I'll scoot around the bottom. That one there is the probability of producing a pod at a node in the right zone of the plant. So although it spends a lot of time in the reproductive phase, it makes a lot of nodes in that phase, it doesn't necessarily convert them all to pods. Seems like a reasonable trade-off. You can't do everything. But this particular one is the highest yielding one. So over here, I've got my yield. If I just come here and put them all back on, it's very hard to make it unselect. You can see that one there, I've broken the yield down into lateral, on the branches, which we'll have a look at in a minute, and on the main stem. You can see that one and the other winning ones, if you like, all had a similar strategy. They kind of, to one degree or another, they spent longer in the flowering zone and were willing to trade off with their conversion rates. But there's actually a lot more here than I can talk to you about right now. That there's a lot of interesting trade-offs and connections here between whether you produce branches, with what probability, where you produce them, at what rate you decide a branch will become fertile, how many peas you have in a pod, the weight of each pea. You're starting to look at different trade-offs along the whole decision process in the time series. I want to show you one more thing here, which is at the moment, these are grouped by sort of somewhat standardized genetic variants on the left here. It's a little complicated because of nonlinear and link functions and things like that. But what I did is by clicking this button, I say, oh, Claude made this interactive for me. But what I did mathematically is say, well, what if I weighted those by their yield contribution? That is, you would have a high value here if you both had high genetic variance and you had high leverage on yield. Obviously, those are more interesting targets than something that has genetic variance but doesn't impact on yield. And so now the candidate traits are clearer here in terms of those that are further away from zero. And we can still look to see how these sort of trade-offs start to work as we move through the different ones. I'm going to have to leave that there for that slide so that we can keep going. So the visualization aspect is so helpful, I think. Like for that previous slide, this one here, I could in principle code such a thing up, but it takes ages getting these things right. It's pretty fiddly. It's so nice to just say, I've got the model fitted. These are just show me a plot like this so that I can interact with it, because I learn a lot about the model. I can see where it's wrong more easily, which is one of the most important things when you start visualizing. So another one we did was to actually simulate the growth of the plant itself. We had pretty comprehensive time series things. Why not just sort of simulate the growth? So I asked it to do that, and I'll just pick the first one in the list here. And so that shows it growing. It shows where the branches were, how long they were, where the flowers were, where the abortions were. And when I showed my biologists this and they started flicking through them, they're all happy and like, hang on a sec, you can't get a branch there in that genotype, and so they're still finding small data entry errors. And I know this is true for everyone, so I don't feel bad saying it about our team. It's hard to get the data absolutely right in the thing, and you want methods to find your errors. But here you can dial up other types of ones so that you can see how they grow, and you can start to see the diversity of genotypes in terms of their branching, heights, the time and speed at which they grow, and the sort of schematics there for the yield. So this is great for science communication as well. Like this is a nice thing you can show people. People nod their head and go, yeah, that looks kind of cool. Oh, it's nice to see. And that really does show us what happened. There's thousands of plants. I mean, we can really see how any one of them grew just by dialing it up. Okay, so where am I? Back to... Yes, this one. That was coded. I think I wrote that paragraph there to make that just a decent paragraph. Not from cold, from this directory where everything is kind of known. It asked me a few questions and then off it went. And then I refined it a few times after that. I tried to make it, give it some confidence. I said, I'm sure you can achieve this. You hear about these tricks and they seem to work, and you think, I feel silly writing this, but I'm going to write it. I'm going to tell you that you're very capable with this. So yeah, it's anyway. I thought I'd be honest and give you the exact thing I typed in with the typos and everything. Of course, you don't need to type that well for AI to understand you. We're at the moment working on a branching model, like more details around the lengths and locations of branches, and sort of conditional on a branch growing low, what will happen to a subsequent one. So sort of building that into the sort of bigger picture of the statistical model that we've got coming along here. Also been using all this is Bayesian inference. It's pretty quick for this starter set, maybe 5 or 10 minutes at the moment, with the branching, which is more refined, a few hours. But I'm also working on another project on much faster GPU accelerated Bayesian inference. And this is great, following on from the previous talk, looking at kind of, factor analytic methods and grouped factor analytic methods and covariances between them and similar such things, especially where you've got G matrices and phylogenetic sort of structures associated with those latent effects. Very technical work, but very exciting at the moment. A few reflections to finish off. I think I could have written this model without AI. I don't know if I would have, or could I have justified the time? I mean, in the sense that I work on so many projects and these guys came along, so could you give us a hand? I said, oh, well, let's have a meeting. We'll have a chat. And then it felt much more achievable to think, I think I can give a couple hours to this project a week and make some really good progress. And that's what's happened. It took a couple of months. Just one morning a week, I'd work on this project, and so much more achievable. I feel a lot more ambitious with AI. I'll definitely say that. I talked a bit about the benefits, the visualization and the wrangling and the science communication, and I guess coming back to the beginning, we opted here Bayesian inference. I guess it's what we know. We saw earlier today that it's actually still performing well in certain areas. And of course, in the future, I'd expect there's going to be much more hybrids between the sorts of things neural networks can do, which are incredible, and us sort of learning to refine their architectures or extend their architectures to really harness the sorts of data we have and the sorts of systems we're modeling. So it's that's an interesting space. I think I've shared most of my tips already. It's good to get it to show its own work. Oh, my favorite one is write me a tutorial as if I'm a student. It just helps me understand things a lot better. And like any tool, you just have to use it a lot to get good at it. Like you just have to. It's a weird beast. So thank you very much for listening today. These are my collaborators. I'm with the Center of Excellence down in Yates.
That is bonkers cool. And I think it really highlights the importance and the benefits of collaboration. So you wouldn't have been able to do that without the biologists. They would have been able to do it without you.
I don't even know what a P is without those guys.
Any questions for Luke? Yeah, Christine. Honestly, bonkers cool.
Yeah, just thanks, Luke. That was lovely. Just a reflection on the future again. Measuring phenotypes is really important. We heard yesterday about 3D scanning to do that and so on. Do you think this sort of trait interaction stuff done in this manner could be used to sort of prioritize traits to be measured? I don't know, Katie, probably your point here, but if you're confident that the trait A always affects trait B, and trait B is expensive to measure, that's the one you really want, but trait A is cheap. And could you use it for quantitative? How many plants I can afford? I've got this amount of budget. You could imagine doing this whole economic analysis on this.
Yeah, that's a good question. We come across that a lot in ecology. Like we think of plant traits and like hydraulics and vulnerabilities to drought and things. Some traits are so hard to measure; they're destructive on the plant. That you can find good correlates, through analyses like this, and a lot of multi-trait phylogenetic comparative analysis, and you can lean on closely related species or partial measurements or incomplete data, and of course, if you find a cheap to measure trait, yeah, definitely. Also, like that earlier plot with the leverage, if you know it's a high leverage trait, it's more worth the effort. And if you know that in advance, you'll invest more money and time in that one. So, yeah, definitely.
Anyone else? Got off easy. Did I see your hand there? Yeah. Yes. At the time. Yeah, go for it. We make our own rules.
There's a minute left according to my schedule.
This is an easy question. Just the visualizations are so cool, Luke. And I mean, years meeting at a whiteboard, trying to talk across disciplines, using lots of hand gestures. It's still an investment in tokens, and you still highly recommend that now with most of your projects when you're working as the modeler for a group of people in the applied domain. And
yeah, the tokens are cheap. I mean, I have $100 a month subscription, but I never use it all. And I work on heaps of projects. I work it all the time. Like it does months and months of work every week. Like the economics are pretty obvious. And for the work I do,
interactive as well, and you can. It's better than generating all the figures and then and letting them interact with the results and.
But we still got, if we had photos of our whiteboards, we still got in there and pointed and talked and things because we were building our own context in the beginning. So it's all good.
Your unit metric box. Yeah. Yeah. Cool.
Yeah.
Very cool.
Great. Thank you.
Right. Let's thank Luke again.